Hi, my name is Abby, and in today's video, we will be looking at different point mutations. We'll be starting with this strand of coding DNA. We'll begin by translating the DNA into mRNA by looking at each base pair. Given that this is a coding strand, we can easily translate by shifting the thymines of DNA into uracils of RNA, giving us this sequence. From here, we can look at our universal coding dictionary to figure out the reading frame and protein products. First, we need to find our start codon. Here we have found the methine. Lucky for us, it is at the beginning of the sequence with the AUG codon. Now we can set our reading frame. Using the dictionary again, we can determine our amino acids. Point mutations are mutations that change one nucleotide base. We'll be looking at 4 types of point mutations. Missense refers to a point mutation that changes the amino acid coded for. This would be like having a vanilla ice cream machine suddenly start making chocolate milk. The produced protein is different and usually does not function like it is supposed to. We can create this by changing the AAC codon into AAA. This would change the coded amino acid from asparagine to lysine. In this case, since we are switching a C to an A, this would be a transversion since it's a pyrimidine to a purine. A nonsense point mutation can be particularly bad as it changes an amino acid into a stop codon. This can shorten protein chains and make them change function or stop working. This would be like pushing a button on a vending machine, expecting to get a snack, and it just turning off. We can produce this mutation if we change the codon UGG into UGA, changing what would be tryptophan into a stop codon. Guanine and adenine are both purines, so this mutation would be a transition. One of the most common results that happens in mutations is called a silent mutation. These happen when the mutation does not change the amino acid produced, so it appears as if nothing has changed. This phenomenon occurs due to base pair wobble on the 5' end of the anticodon, which allows different bases to pair, letting multiple codons code for the same amino acid. We can show this by changing the codon GCU, which codes for alanine, into GCC, which still codes for alanine, and would cause no change in protein products. As this is changing a pyrimidine into another pyrimidine, this would be a transition. The last of the point mutations we will talk about is the readthrough, which turns a stop codon into an amino acid, making the protein product include amino acids it was not meant to have. This would be like if you wanted to pause a game of Tetris, but the blocks just kept coming. We could get this mutation by changing the stop codon UGA into CGA, which codes for arginine. Since this changes from a U to a C, it would be a transition.